All right, we've hit 12:03 and we're starting to level off, so I guess we'll get going. So, hello everybody, and welcome to this week's SBGrid webinar series. Today we have two presenters who are going to talk about some viral-related stuff, which is always interesting, and particularly interesting with the current events over the past few months. If you have questions, you can send them to me or one of the other hosts in chat, and we'll moderate those at the end. And so for today, as I said, we have two speakers, both from the Acharya lab at Duke University School of Medicine. So thank you for joining us. Our second speaker of the day, Kartik Manne, who's from the same lab, the same institution, and
you can see my slides, right?
I can see your slides. More vaccines, different virus.
Yeah, another pandemic, right? So my name is Kartik Manne, and I'm a postdoctoral associate at Priyamvada Acharya's lab. And the title of my talk is the structural studies of CH235 lineage of antibodies for HIV-1 vaccine design. So I want to switch gears from one pandemic that Sophie talked about to another current pandemic, that is the HIV. And according to the World Health Organization, there are about a total of 38 million people living with HIV as of 2019, with 1.7 million people newly infected last year, and around 690,000 total HIV-related deaths in that 1 year. It is also estimated that approximately 1.2 million people in the US are living with HIV today. So for all these reasons, I think we need a vaccine to prevent HIV. To give a brief background, the human immunodeficiency virus uses protein spikes embedded in its envelope to enter the host cells. And this envelope, which is showed here in a zoomed panel, is in a trimeric form and is composed of receptor attachment subunit called GP120, colored here in purple, and the fusion subunit called GP41, colored here in green. And the CD4 binding site is in yellow, which is an important receptor site for the CD4 cells. Unfortunately, the HIV-1 envelopes have conserved and accessible regions, and the naturally elicited antibodies target these vulnerable sites to neutralize the virus. In this slide, I'm showing each of these sites of vulnerability, and let me point out that this is the viral membrane, and that's the envelope. In gray are the glycans that's shielding the envelope, and the colored regions are the regions of sites of vulnerability. And mentioned here, tabulated here, are the antibodies from those classes of vulnerable sites that were isolated from infected individuals. And the CD4 binding site, which is the site of initial contact of the CD4 receptor, is important site that blocks the receptor from engaging the virus. And the CH235 lineage of antibodies, the ones that I'm interested in, target this CD4 binding site for neutralization. But the problem is, typically these antibodies take many years to develop and mature. And the solution to that is to teach the immune system to rapidly generate these antibodies when faced with an infection. Here at DHVI, we are lucky to follow patients and isolate antibodies from early stages of infection to advanced stages. So what we do is follow the isolated antibodies at different stages of infection. In this figure, I'm showing what we show are the neutralization dendrograms colored by its neutralization breadth. The black lines represent no neutralization, and the red lines represents highest neutralization breadth, and rest of the colors fall in between. The CH235 lineage of antibodies isolated early in the infection called CH235.UCA, which is short for unmutated common ancestor, showed weak neutralization. And here you can see, the dendrogram shows no neutralization. The lines are all black. And as we followed the same infected individual and isolated antibodies over time, the mature antibody, which was isolated close to 6 years from infection, which is called the CH235.12, had broad neutralization, and you can see red lines, which indicates broad neutralization compared to the early CH235 lineage member. So the central question we are asking here is, what is the structural basis for the increase in this neutralization breadth? So again, to remind or to reiterate, the early CH235 lineage member, which is called the UCA, has weak neutralization, and it develops into a mature antibody called CH235.12, which shows broad neutralization. And we're asking, what's the structural basis for this? And to give you the answer straight away, we found that the glycan 197 is a key barrier that the CH235 antibodies need to deal with. And to understand the structural basis for the glycan 197, we asked the questions, how does the glycan 197 bind to the early member of CH235, which is CH235.UCA, and how does that compare to the mature CH235 lineage member, which is CH235.12? Next, we asked the question, how does the CH235 lineage gain the ability to accommodate this glycan of interest, which is glycan 197? And the exciting part is, how do the vaccine elicited antibodies interact with this glycan 197? And here in our lab, we followed the this workflow for the structural studies, where I would transfect the plasmids into mammalian cells, and followed by their corresponding IgG and Fab purification. I would use the purified Fabs to make complexes with the envelope, check for their binding, and determine their structures using cryo-electron microscopy. I'm also trying to solve the structures of the unbound Fabs using X-ray crystallography. So here in our lab, we are employing hybrid methods to solve the structures and answer some of these important questions. And let me give you a little bit more background about the methods for the cryo-EM structure determination. The antibody Fab and the envelope complex were made by mixing the purified samples at 6-fold molar excess of the Fabs. And the complex mixture was deposited onto a copper grid, plunged frozen into a liquid ethane using LECA. And the cryo-EM data was collected at Titan Cryos here at Duke. And the data processing was done using CryoSpark, where we selected these 2D class averages and built an ab initio model where we can see 3 Fabs bound to the envelope with a C3 symmetry. And then in this figure, I'm showing the model of CH235.12 Fab bound to the envelope. And that's fitted into the refined map at a resolution of 3.7 angstroms. And then we compared the CH235.12, which is the mature antibody of the CH235 lineage member, and the CH235.UCA bound to the envelope. When the GP120 subunit, which is the receptor binding subunit of the envelopes, were superimposed, we discovered that both these antibodies bind at a similar angle of approach. Basically, they're oriented in the same way on top of the envelope. So this reveals that the CH235 lineage finds its angle of approach early on in its development. And looking closely at the binding interface, similar to the CH235.UCA, which is the early member, the mature member Fab bound to the envelope using the complementarity determining region on the heavy chain called the CDR-H2, and that makes key interactions with the GP120 CD4 binding loop colored here in pink. In addition to the glycans, studies were done to show that two loops at the interface known as loop D, colored here in yellow, and loop V5, colored in orange, pose barriers to the CH235 binding. So we looked at these loops interacting with the antibody, and when we made two mutations, one on loop D, where we mutated the N279 residue to lysine, and the other mutation, which is G458 to tyrosine on loop 5, this increased binding to the early CH235 lineage member, which is CH235.UCA, and that was needed for the UCA to bind to the envelope. However, we have observed that the mature member, which is CH235.12, does not need these mutations for binding to the envelope. And the key difference in the binding of CH235.UCA and the mature member, that is CH235.12, is the glycan 197, the glycan we are interested in, right? So here on the left, you can see that glycan 197 is, we were able to see good density for glycan 197 and build the glycan with pretty good confidence. And that was missing in the earlier member, which is CH235.UCA bound to the envelope. So this glycan was disordered in this complex. We have also observed that the glycan 197 interacts with the CH235.12 heavy chain residues, tyrosine 70 and tyrosine 79. They stack against the sugar rings and helps in the binding. And most interestingly, we've observed a residue at position 19, which is threonine in case of CH235.12. And this was, present in the region of glycan 197 interaction. We were interested in this residue because in the UCA, which is the early member, this residue 19 is a lysine. And when we modeled the lysine 19 residue into the CH235 bound to the envelope, we found that the lysine would clash with the glycan 197. So we hypothesized that the K19T mutation is necessary to accommodate the glycan 197, allowing better binding to the antibody to the envelope. And currently we are investigating the binding and structures of HIV-1 envelopes in complex with mutant CH235.12 and CH235.UCA, where basically what I'm doing is I'm taking the early member, which is the CH235.UCA, which doesn't bind to the envelope very well. I mean, which has the lysine in residue position 19, and I'm mutating that to a threonine, and we expect to see a higher binding in this mutant. And we are also investigating the binding, and then we would like to study the structures of the CH235.12, where I mutate the threonine 19 residue and revert it to the UCA residue, which is lysine. And we expect to see a decrease in binding of this mutant to the envelope. So to summarize the first question, again, the first question we asked was, like, how does the glycan 197 bind to CH235.UCA compared to the mature member CH235.12? And we found that the glycan 197 UCA is disordered. And in the mature member, the glycan is ordered and interacts with the antibody heavy chain. So the second question we asked was, how does the CH235 lineage gain ability to accommodate the glycan 197? And in this slide, I'm showing you how the CH235 develops from UCA to the mature member CH235.12. And as the time progressed from week 14 to week 323, colors change from cyan or teal to purple. And the point I want to make in this slide is that the CH235 lineage developed the ability to deal with the glycan 197 early on in its development. And we hypothesized that it does that by undergoing this key mutation, K19T mutation, early on in the lineage. And so we wanted to look at one of the early lineage members, which is called the CH235.I60. And we went ahead and solved the structure of this intermediate CH235I60 in complex with the envelope to a resolution of about 4.1 angstrom using cryo-electron microscopy again. And as we can see here in the ab initio model, we were able to see 3 Fabs bound to the envelope. And the resolution, like I mentioned, was about 4.1 angstrom. And then I looked at the local resolution of the refined maps and have colored the map by the local resolutions, where blue represents better resolution, which is about 3.5 angstroms, and the red represents low resolution, which is about 7.5 angstroms. And the region of interest, which is right here and here, is the glycan 197, and that has a pretty decent resolution of 3.5 angstroms, which allowed us to build the glycans with confidence. And we were able to build the glycan 197 for the intermediate, which is CH235.I60 bound to the envelope. And this part of the structure looks very similar to CH235.12, which is the mature antibody. So the intermediate, which is the early on intermediate of the CH235 lineage, has the glycan 197 accommodated, which is similar to the mature CH235 lineage member. And therefore, it's safe to state that the goal of accommodating the glycan 197 is met early during lineage development by making this key K19T mutation. Again, the question we asked was, how does the CH235 lineage gain ability to accommodate glycan 197? And we found that the goal of accommodating the glycan is met early on during lineage development by making the key mutation, which is K19T. And the final question, very exciting question we asked was, how do vaccine elicited antibodies interact with glycan 197? And here at DHVI, the investigators carried out a study where the animal models were vaccinated with engineered HIV-1 immunogens that generated antibodies which exhibited neutralization activity similar to that of the intermediate stage antibodies that were isolated from humans. So these are the antibodies that were generated by the animal models upon vaccine. So we elicited these antibodies from vaccination of the animal models, and they look very similar to the human, the antibodies developed by humans nascently. And one of the vaccines that we were interested in that were elicited that were vaccine elicited from the mouse model is the CH235.mu2, which was shown to be CH235-like. And this vaccine picked up the K19T mutation that we were interested in. And we wanted to see how it interacts with the HIV-1 envelope structurally. So we solved the structure of this again. We solved the CH235-mu2, which is the vaccine elicited antibody, similar to CH235, in complex with the HIV-1 envelope using Cryo-EM again. And we could see 3 Fabs bound to the envelope with a 3-fold symmetry. And lo and behold, we found the glycan 197 interaction with the vaccine elicited antibody heavy chain, which is similar to CH235, which is the mature bound structure. And like I mentioned, the K19T mutation that was adopted by these vaccine elicited antibodies were observed in the structure, which basically accommodate the N197 glycan. And if this was a lysine, like I showed in the previous image, that would have been clashing with the glycan. So to summarize this question, which we asked was, how do the vaccine elicited antibodies interact with glycan 197? And the answer we found was the vaccine elicited antibodies, CH235-mu2, which is CH235-like, has acquired the mutations, the K19T mutation, for example, needed to interact with the glycan 197. So in conclusion, I'm repeating myself, but I want to point out 3 main points from my presentation. That is, glycan 197 in the early lineage member, the CH235-UCA complex, is disordered, whereas in the CH235.12, which is the mature CH235 lineage member, the glycan is ordered and interacts with the antibody heavy chain. The goal of accommodating the glycan 197 is met early during the lineage development by making the K19T mutation, as we have shown in the intermediate complex structure CH235.I60. And fascinatingly, the vaccine elicited antibody CH235.mu2 has acquired the mutations needed to interact with the glycan 197. And our structures say that we are on track with the vaccine, and we are eliciting antibodies using the current vaccination strategy. So we are on the right track towards a vaccine for the HIV, I would say. And with that, I would like to thank my PI and DHVI Structural Biology Lab Director, Division Director, Priyamvada Acharya. And I would like to acknowledge all the collaborators at DHVI that were part of this study. And I would like to thank our amazing structural biology team, RJ, Shena, Katarzyna, Megan, Priyamvada, Rory, Sophie, who you heard talking just now, Katayun, and Victoria. And finally, thank you all for attending the talk, and I'll take any questions you may have at this point.
Thank you, Kartik. That was a really interesting talk, and a reminder that just because we have one pandemic in the news doesn't mean that the other pandemics have gone away yet.
Yeah.
So if anybody has questions, feel free to send them to me in chat. But I guess the one question that's possibly well known to people who work with HIV, but what makes glycan 197 such a target for antibody binding? Is it just like a convenient place to bind on, or does it have some more implications for the viral life cycle?
So the accessibility of this glycan in 197. So this glycan is basically a barrier for the antibody, right? So only the antibodies that learn to interact with this and bind to this glycan can actually show some kind of neutralization potency towards the virus. Not many antibodies actually do that. So the CH235 lineage antibodies are some of the few antibodies that actually interact with, or as far as I know, very few of the antibodies that actually learn to deal with this glycan hence we are interested in. There are some more from this class which bind to the glycan similarly that are being studied as well.
And do you have any thoughts on if the glycan accommodation occurs relatively early in the antibody lineage, what improves as things go down the line?
So this N190, the glycan 197 binding, like I showed in the phylogenetic tree of the CH235, we have taken the early lineage member, which we call CH235.I60, or which is called CH235.I60. And we have seen that right away compared to the UCA and this early intermediate, we were able to see, the glycan 197 being accommodated. And the biggest reason being the mutation at the 19th residue, K19 to T. And from there to how it progresses to a mature antibody, which is CH235.12, there are a lot of other things happening. This is just one mutation. Actually, Kevin Lee at DHVI has pointed out all the mutations going on in the lineage of antibodies, and there are several, and some of them have relevance in making the antibody better, binding to the envelope better, and helps in neutralization, and some of them are irrelevant. But to answer your question, there are many more mutations going on, starting from the early on antibody to the mature.
So some of those mutations are probably analogous to drift, but some of them probably have some functional consequence. I guess a non-immunologist would assume that that's probably better binding or something like that. Could you comment on that?
Yeah, some of them definitely have the functional importance. Some of these mutations, like for example, the K19T is functionally relevant structurally too, we showed. But there are other mutations, like for example, there's a mutation at the 50th residue where it mutates from isoleucine to tryptophan, I50 to W. That eliminates, the figure where I showed there was a mutation G458Y on loop D that helped binding to of the envelope to the UCA. Because of the I50W mutation in one of the intermediates, CH235 intermediates, that eliminates that probably, this is all hypothesis, but we have to look at it structurally as well. But that probably eliminates the need for the immunogen to have this mutation G458Y, because it's lying in the same area over there. Yeah, but that's a big question we are all after.
Yeah, there are always more questions than answers, it seems.
Yeah, it's
so
all what is this doing? It's $1 million question.
So we have another question that I might know the answer to, but you're the expert. Does COVID have glycans?
Yeah.
Yep.
Yeah.
That's what I would have thought, but I am not the virologist. Yep. So with that, I guess it looks like if anybody, last call for questions, and if not, I'd like to once again thank both of our speakers. So Sophie and Kartik, thank you for taking time out of your research to speak with us today and tell us about what you're doing. And thank you for 2 very great talks. And everybody in the audience, thank you for attending.
Thank you.